On this project, we will be learning about the cytoskeleton. We will now talk about the three main elements of the cytoskeleton. Now, the first type that we're going to talk about today are called intermediate filaments. Now, let me draw a more visual representation of what these filaments look like. The black structure surrounding the main drawing is supposed to represent the outer part of the cell, or the cell wall, while the brown circular structure in the center is supposed to represent the cell nucleus. And as you can see, the blue squiggly looking lines coming in from the cell wall are the intermediate filaments. And as you can see, they are spread out throughout the entire cell and are rather thick compared to other filaments we will see later. Now let's discuss some of the features of intermediate filaments. The first one I would like to mention is the mechanical strength, and this is due to their fibrous nature and rope-like construction, which allows them to have a high tensile strength, and this allows the cell to maintain a shape and basically give it the ability to withstand pressure and be able to hold itself up. They are also the most prominent type of filament in the cell and the most diverse, as it is located throughout most of the cell. Another feature is that they are grouped into 4 different classes. One other feature of intermediate filaments is that they are grouped into 4 different classes. This can be described by keratin filaments, vimentin-related filaments, and nuclear lamins. The next part of the cell cytoskeleton I would like to talk about are the microtubules. Now let's provide a more visual representation of this. Right above the nucleus we have the centrosome, in which the microtubules then come out of that and then radiate throughout the cell. Now let's talk about some of the features of microtubules. The first one is that they are very stiff, and this gives them rigidity and much more structure within the cell. The second feature that is unique to these is that they come out of that centrosome right in front of the nucleus. And the third most well-known feature of these microtubules is their help in cell division. These can form structures that will help pull apart various elements within the nucleus and assist essentially in cell division. Now let's give a more visual representation for the third element of the cytoskeleton that we will talk about. Actin filaments, as you can see here, drawn in red around the wall of the cell and up into those little crevices, are much thinner and much more flexible elements of the cytoskeleton than the other 2 elements that were mentioned in this video. Now on to some of the features of actin filaments. The main feature that I mentioned before is that they are much thinner and much more flexible than the other elements of the cytoskeleton, and this allows for some unique things. The first one is that they are made of polymers of the protein actin. This is kind of obvious, hence the name. Now, the other feature that is crucial to actin filaments is that they give the cell its ability to move, and this means that it can crawl and interact with its environment. This also allows it to interact with other cells in its environment. I will now turn the project over to my partner, who will talk about how the cytoskeleton and specific information about it can be used in the medical world today.
Hey guys, in regard to the medical world, we will be talking about microtubules. as well as the two medications paclitaxel and colchicine. Paclitaxel, also known as taxol, and colchicine can both be used to treat human cancers. Although different in some ways, they both use very similar methods to perform their job. Cancerous cells are characterized by uncontrollable and unregulated cell division. Chemotherapy fights cancer by trying to stop this cell division. Taxol and colchicine are both classified as antimitotic drugs. These types of drugs either stabilize or destabilize microtubules to inactivate or destroy the mitotic spindles that play a role in cell division. Microtubules are microscopic hollow tubes found in eukaryotic cells that play a role in the structure and motor functions of the cell. Microtubules are made up of subunits of tubulin. Tubulin is a dimer made by two globular proteins known as an alpha and a beta tubulin. Tubulin organizes into linear chains known as protofilaments that come together in groups of 13 to form a microtubule. It is also important to note that the presence of an alpha and a beta tubulin creates a structural polarity in the microtubule, with one end showing beta tubulin being the plus end and one end showing alpha tubulin being the minus end. Polarity is important in the guidance of directional intercellular transport, as well as the microtubule's role in cell division. Microtubules are important in cell division because they reassemble into mitotic spindles. Mitotic spindles separate the replicated chromosomes to opposite sides when two daughter cells are being created. Taxol is a member of the chemotherapy drug group known as taxanes, a subunit of plant alkaloids. Taxanes fight cancer by binding tightly to the microtubules and preventing the loss of its subunits, the tubulin. When this occurs, the microtubule will keep growing because it will continue to gain subunits while losing none. By stabilizing the microtubule and preventing disassembly, taxol causes the mitotic spindles to fail to function. Colchicine is used mainly to treat gout and familial Mediterranean fever. However, it shares a similarity to taxol because it is also a member of the alkaloid plant family. However, colchicine acts differently to fight cancer. Colchicine binds tightly to free tubulin dimers and prevents their polymerization into microtubules. This leads to the disappearance of the mitotic spindles and the stalling of cell mitosis. By altering the microtubules and thus the mitotic spindles, taxol and colchicine stop the rapid division of cancer cells. This goes to show one of the many ways that the cytoskeleton plays a role in the human body and how medicine uses our knowledge of it. Thank you for watching this video, and we hope you learned something.